Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PIcc is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of the discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
Screen share, please. Hello, welcome everyone to the PIcc Steering Committee meeting. It's September 29th, 2021, Earth. And we have a great agenda, and let us just get started here. I'll show you a quick overview of the agenda here. So there's, a lot of news, and if I'm informed correctly, Ola will paste the agenda in the chat so everyone has all the links and all the resources. I don't know if Carla and or other folks are available from PIcc. I think they may be, but let's get started. So the first and probably most significant accomplishment here is that a company has received the first ever FDA approval for an eye product in digital pathology. I think probably most of you have heard about it. If not, please take a look. This is obviously no product placement, but simply a thank you also to some of the reviewers across over in the FDA, because I presume there was a lot of technical detail that went into this, and I think it starts sort of unlocking some of the potential. So I presume that at least for this particular application, which was prostate Gleason grading, we have now a predicate device, and thereby, we have some sort of performance standard to measure other things against. So I think that is definitely an accomplishment for the company, their regulatory science team, but also for the FDA. So I just thought I wanted to point that out. Just see here. Is there, I don't know, I think Carla was at least at some point there. I don't know if she wants to say a few words or if other people have comments on this. Has someone tried this tool before? Don't know. Maybe you can put it in the chat. Well, in any case, I think as soon as all the documentation will be out, which I think will be in a few weeks, I think there's definitely a very interesting document to take a look at. So we will probably do this with the AI group to really look through how this, I think in this case, a classifier was assessed, how the performance was measured against human. I think this will be extremely insightful And, I think maybe if Carla joins later, she can say a few words. How much of this, currently I think it's in press release stage, but I presume that session will also be of interest to, some of the reviewers on the FDA side to hear what we're thinking about, for other tools that are more in the quantification space, etc. So, I don't know, Stephen and Matt and I will probably also take into account what's in there for the AI rubric that we're building. Questions about this? Probably not, but definitely a great accomplishment. Next, if you haven't seen it, Ulla, maybe you can add the link as well. I know it's in the agenda, but just for those who may just join. The FDA has launched an artificial intelligence and machine learning enabled medical device list. This is a screenshot, and the link you will find in your in the chat of this meeting right now. It's extremely helpful. If you scroll through, I read through many of those. The documentation is extremely intricate. Interestingly, the first ML, let's say, enabled medical device was actually a tumor of unknown origin classifier, interestingly. So if you search through that, that's something very interesting. And if you look at, some of the, combinations of, let's just call it device, company, and use cases, I think what I believe is very interesting for this group is to just read through the specific intended use wordings, because I think that is something that is incredibly important to get that right and to also gain experience from a, just more of a device developers. And I know there's a lot of algorithm developers here. So I think that is definitely very helpful. And so with that, I don't know if someone's on here. I know Brandon didn't have time, but maybe Kate, I see Ed is also on the call. If you know some background on this list or, but I presume it's intended to be a little more transparent on what's out there, right?
Here, maybe I can just say words. I think it's just more broad that we're trying to get as transparent as we can and try and help really give some ideas to people as well, because when you know what has gone through and been approved and cleared, that in itself can actually spur ideas across the whole community. And that was one of the reasons that I know that Jeff Shuren, our Centre Director, wanted to push ahead with this. So I think it just makes sense. It's not like there's a proprietary information here or anything like that. But I think it's quite exciting that for me, what was particularly telling was that the dates aren't all 2019, 2020 and 2021. We've been at this for years. We broadly, not me, of course, but the community as a whole. This is nothing new to us. And it's something that I think isn't always apparent across the rest of the agency as well, but our reviewers and our incredible people at FDA have been dealing with this built into devices as well as infrastructure tools for decades, really.
Yep.
And it's just really blossoming most recently. So just a couple of bits of context from me.
Awesome. Thank you, Ed. That's really good to hear. I just wanted to point out this sort of first, at least, this is totally personal. I don't know. I haven't looked into like the really in-depth, but there may be other things that, there are some earlier approvals, but this sort of tissue-based AI or machine learning, this was from 2012. So clearly the FDA has looked at this and, looked at, sort of the performance assessment. So I just wanted to, point that out. If you haven't seen it, this will definitely get you hopefully some, additional ideas and or, components to your research and or, sort of, I don't know, to spurt your developments and, find other people and or experts. All right, the next 3 slides are 3 relevant papers that I just wanted to briefly highlight. The first one is I would call it a summary work, opinion piece, and implementation plan written by, these authors, very accomplished regulatory science experts. Some of them are known very well to this group. The senior author, Laxman Ramamurthy, is sitting here. And looking across the ocean, because this is about the in vitro diagnostic regulation in Europe, the IVDR, just in case you wonder why is there a cover like that on a regulatory science paper. So the publication here in the Journal of Precision Medicine, which is really a high impact and an important journal in this realm, is sort of outlining some of the specific regulatory changes that are imposed here. And I wanted to quickly highlight one of the things that I believe is really, I don't know, the best part of this, which is a proposal for the phased implementation of this. So this is the whole content and a very good overview. So if you haven't seen this, please read it. But this is clearly sort of throwing the ball across to the regulator to say this would be an idea on how to implement the IVDR and what that would entail. So with relatively concrete numbers and classes. So why I thought this would be of interest is that, of course, we have a potential change of laboratory developed test regulation pending, and then we will come to see whether the Valid Act would make sense and how that could be implemented. So I think there's definitely some parallels drawn in this article. So I wanted to definitely give a shout out to the group. It's a great paper, 5 accomplished regulatory science experts who got together, and if I'm seeing this right, the paper is now pasted in the chat, so please take a look. The second paper, and one of the senior authors is, I believe, also on the call, Stephen Yip. This is a paper by Adrian Levine, and this is, I think, posted previously on bioRxives, but now published in the Journal of Pathology, which is also, a high-impact journal. Is called Synthesis of Diagnostic Quality Cancer Pathology Images by a GAN, so Generative Adversarial Network. And Stephen, I'm putting you on the spot here. Do you want to say a few words, what you have accomplished here?
Well,
Since
Ali, the senior author, is here as well, but he's
perfect.
I don't know whether he can speak or not.
Oh, got it. Yeah, so if possible, it would be great. If not, I can just try to summarize it real quick.
So if Ali can't speak, basically we use TCGA images for, I think, low-grade glioma, kidney, and one other tumor, but also use a variety of glass-based image of different ovarian subtypes as well to generate synthetic images using very typical GAN network. And also we presented that as a way to challenge pathologists by presenting side-by-side a synthetic image versus a real image, and they have to say which one is real, which is not real. And I think we have some interesting results. So basically here you see match synthetic as well as real images. Obviously with GAN, sometimes you do have some image abnormalities that you can easily see is fake. But I think in a way this helped to, at first I thought this is kind of novel and kind of cool in a way, but then you have to think about the practical implication for it, and some of which might be, we can actually use it to generate synthetic image of rare tumors. For example, if you want to train a network to identify different types of rare tumors, then you might not have sufficient glass slide, and you might want to use that. And we actually have, and Ali have actually done it, to use these synthetically generated rare tumor images to train a network and able to show that they actually work quite well. I think the other potential application is to the privacy thing. It's like you can actually generate synthetic images that is not linked to any patient because these are completely novel. So these are two things that we think is quite useful, but I'm sure that other people can think of potential application as well.
Perfect. Hey, thank you so much for the ad hoc summary. So I think especially the last part, I know we have a privacy and an information blocking sub-project. So having basically a ground truth that's computer generated is, some people consider that a breakthrough, and I truly think that is amazing. So congrats on the great paper, and we were a little late with catching this, but I think it is applicable to some of our questions and projects here. The third one, I won't say a lot about it, but I definitely wanted to mention it. It comes from a group here at MGH. Heather Marble is known to some of you, and some of the other people you may recognize as well. I just wanted to briefly mention that this is a paper outlining some of the temporary regulatory deviations that happened during the COVID pandemic. I don't know if you heard about it, but it's a coronavirus, it was like a big deal. In any case, so the questions that we have here were, what are the relevant changes when it comes to pending LDT regulation by the FDA? And the two things that I thought I point out really quickly here is, the LDT law ongoing basically since the 70s. There was a Cooper committee report. So this timeline was suddenly changed with the pandemic and basically required, I think that the enforcement discretion, as many times previously, was overridden, and the FDA asked to see some of the validation reports or performance establishing experiments. And then a little bit later, there was basically a labeling update study to assess the performance. And what basically came out of this study is a concept on how diagnostic tests and diagnostic procedures and diagnostic services are a concatenation where in the center is an IVD or an LDT, but that the diagnostic quality depends more on how these different diagnostic components are interrelated. And then, of course, to get a diagnostic test to a patient is sort of a maze. So I thought that it would be, interesting to just point that out. One other thing, quickly going back, and I think, Stephen, you had sent me the link. I apologize that that's not in the agenda. But if you click on the link, you can test this synthetic GAN network and see whether you can recognize traditional histology or artificial histology. So, quick summary again, one IVDR paper, a synthetic or artificial histology paper, and a regulatory paper on LDT were sort of the relevant papers that we wanted to feature this time that hopefully have some relevance to some of the people. Questions about any of this? Stephen, is the GAN code available? Did you release that, or GitHub?
Should be available.
Awesome.
Ali can probably best answer that. He's our computational guru.
Awesome.
Yeah, and I think it should be available. It should be in GitHub.
Yeah, it should be.
Fantastic. Yeah. All right. Next up are the work group updates, and I think we have two updates, one from the ML and AI model creation group and one from the truthing and ground truth and validation group. So I think we start with the AI group. Stephen, can I hand that over to you?
Yeah, can I share my screen? There we go. Okay, so two things that I just wanted to briefly mention that would be of interest here. One is a shout out for a DPA webinar that's happening tomorrow. So validation of AI-based tools in digital computational pathology. I thought that might be of interest if you haven't registered for that already. The other topic or workshop that I wanted to bring up was this public workshop from the FDA. They had actually solicited for participation on this date for this topic, and myself and a couple others submitted a request on behalf of this consortia to talk about the things that we've been doing, but unfortunately it was declined just because the schedule is just so busy anyway. But it looks like it's going to be a fairly informative discussion, and so I'd encourage participation for that if you could go as well. I'm sure we'll get a word in somewhere. Just with respect to other updates regarding the rubric, that medical device discussion the transparency actually kind of gave us a little insight into how we wanted to frame our paper discussion. In addition to just this rubric or this decision tree, we're trying to figure out a way of telling a story that would be relevant and appropriate. And Joe had pointed out that when we look at some of the FDA documentations, there's usually a picture, and then there's a very small box that says model development and model validation. And so we thought we'd actually take that as kind of the focus of what we wanted to do in terms of doing the translation, because as we've mentioned before, when we asked the trainee group to give us a list of things that you would like to be able to do with AI and whole slide imaging, what the first thing that we realized that we had to do was translate that into what sort of AI speak do I need to accomplish to get to those clinical goals. And so by sharing this vernacular between the two different groups, we think that is what's going to have value and increase transparency and increase understanding, therefore. So that's kind of the focus of where we are now.
Perfect. That's questions to Stephen? Okay, let me just share my screen again. So the next update is from the Truthing and Validation group. This is an updated document. So Kate, do you want to quickly outline your updates?
Can do. Thanks,
awesome.
Joe.
Thank you.
And also thanks, Stephen, for mentioning the FDA AI machine learning workshop. I had forgotten to include that as a FYI for the group. 3 items that are of potential interest is that Brandon recently gave a recorded presentation to the PIcc community as part of Pathology Horizons. And part of his presentation touched on a short discussion about algorithm change control plans and updated thinking in regards to them. So that might be of interest. This past Monday, I was able to participate as part of a panel with the 2021 Computational Pathology Workshop, which is part of the ongoing All This Week International Conference on Medical Image Computing and Computer-Assisted Investigations. And all of the proceedings have been recorded from the workshop. So not only are the proceedings documents available, but also the panel discussions and the live Q&A sessions are available to anyone who registers for that conference belatedly. And finally, Dr. Victor Garcia, who is on the line and is a new fellow, relatively new fellow who joined our group, has been leading some discussions with a subset of our expert pathologists to identify critical instructions for our HCT annotation project. And this will hopefully also lead to less pathologist variability with entire pathology annotation instructions, along with help us specifically with training for this data set. So we've been recording all of these sessions, taking annotated notes, and we're happy to distribute our findings, as well as we're preparing to present the results at Pathology Informatics, as well as a few other venues.
Wow, that's a lot. I think this is fantastic. I wonder if the annotation training would be something also for the trainees group or the trainees work group to help people, especially looking for projects, etc. during training. That would be probably very helpful.
I can definitely share with Julia and Sarah.
Cool, perfect. Questions? I mean, truthing and validation will probably be a continued hot topic for this group, because as soon as the first MDDTs go out, that will be definitely very, very important. Thanks so much. Let's move on. I think the next item on the agenda is an update on the information blocking project. And there was on September 14th by the Office of the National Coordinator for Health Information, or ONC short, a session called What Clinicians and Other Healthcare Providers Need to Know About the Information Blocking Rule, which is part of the 21st Century Cures Act, which went into final effect on April 5. And I wanted to show you the this whole information was curated in this project, and one of the key elements was to establish an organizational policy regarding lab test results. So of course this affects all of medicine and all of the data communication, but these specific subsections were mentioned when it comes to lab test results. And keep in mind, this includes anatomic pathology as well as clinical pathology results, so you still have to like do a little bit of digging. But these three frequently asked questions from January and March, as they were answered and published, have been, curated and copied, but also the original, CFR citations are in here. So you can find this, plus the entire sort of, webinar that was given, I think it was close to an hour or so, is also on the website here. So this is under the information blocking project. So projects and information blocking, in case that applies to your practice. And, there's a lot of information about the 21st Century Cures Act here. So this was a quick shout out regarding the ONC message that was, I think, very helpful. The next section is about 4 of the projects, and the first one is the TILS working group and the PIcc joint survey. Kim, are you presenting this? Or can you? Kim, I think you're on mute.
Thank you. May I share, please? It's not allowing me.
I think Ola can hopefully enable that momentarily.
Should be full. Or Joe, do you want to stop sharing?
Yep.
No worries. Awesome. Okay. So we'll go with here. Can you see my screen?
Yes.
Mm-hmm.
Fantastic. Thank you. So I'll put. Oh, okay. Okay. Right. Okay. Let me see if I can move some of this around. Got too many moving parts. So this is a bit on the PIcc, the joint PIcc TILS regulatory group, TILS RS project, so regulatory science project. And just a reminder, I first of all want to thank Joe for allowing me to give a quick update. The whole point of this is to try to understand what is the best way or the best methods and tools that we sort of like need to have morphological entities such as H&E TILs qualified as biomarkers by regulatory agencies. And so just very briefly, very quickly, I have about 5 things I'll discuss really quickly. 1 are our current members of the project team. 2 is the goals. 3 is the first deliverable, 4 is the next steps, and 5 is the timeline. So here are our experts on our list. Oops. Wow. This is literally the steering team. I'm the project leader with Roberto and Joe as the head members of the steering team. And then we have several wonderful people who've agreed to be part of the group as experts in their respective areas. From academics to biotech to pharma. So the goals of this is literally wanting to try to understand how can we think about things like incorporating TILS into the standard clinical practice, since it's endorsed by the TNBC for triple negative breast cancer by several, big groups like, ESMO and IBCC and the WHO. And so there's a growing interest to have the assessment of the TILs approved by regulatory agencies as a mechanism to make sure that we have some sort of or some level of standardization of the process. And so looking through this, there are two, long-term deliverables that we're interested in. The one being, long-term is, more of a report that contains a summary of evidence supporting the regulatory qualification of TILs as an intrinsic morphology-based biomarker in clinical trials. And the output of this is really going to be a Q submission that we put forward to the FDA. And this is really an opportunity, as you've heard here, I'm quite sure several times, it's really an opportunity to discuss the potential ways that H&E TILs could be used and what types of data would support this approval. And then the short-term deliverable that we have is actually, a series of manuscripts that will be used as data for the Q submission itself. And our first output would be a cancers publication. And really what this is literally a blueprint for the initial Q sub submission on the H&E TILs. And so what's really the point of this? So this first short-term deliverable it's really trying to understand the underlying concept is to consider that the morphological biomarkers, including machine learning biomarker validation on H&E size, could actually really serve as a new paradigm for the FDA and other regulatory agencies around the world. And so in this manuscript, we were trying to really explore, biomarker considerations that could serve as literally things, so factors, summary lexicons that can be included in the submission content for the Q sub itself. And as we were putting through this, going through those wonderful members of experts and the steering team, I came up with seven different sections that this particular publication that's going to be listed in this particular publication. And these 7 sections, I won't go into any detail. All this is already up on the website. Only one of these, as we're trying to put this publication together, have, what would be considered like, enough data that we can actually add to the actual piece of that. And that's, the current research uses, because, there have been several clinical trials over the years that have, included H&E TILs as one of the, outputs and outcomes for the data. But the other things for H&E TILs in terms of technical consideration, the landscaping consideration for controls and the best practice and things like that, we really don't have that data yet, right? And so in order for us to kind of, try to have some sort of data for the paper, and these things, to be very clear, is what the PIcc group is actually working on. It's just that, the studies are not mature enough that we actually have publishable data that we can add into this particular publication. And so with that being said, we're trying to think of a different way for us to collect, data. And so our next step was to think, okay, well, we can actually do a survey. And the purpose of the survey is to really delineate the key questions that we want to ask regarding consideration of H&E TILs as a qualified biomarker. So this will allow us to have consensus and therefore clarity on where the field should focus its time, efforts, and resources. And so there'll be 2 versions of this, and one version will be for the professionals in the field, and the other version would be for the patients and patient advocates, because this is a very important population of people that we really need to understand. And so, what's the projected, timeline for this? So the first draft of the survey itself is actually done, and this is currently being reviewed by Joe and Roberto. And then the second draft I'll send out to that wonderful team of 7 experts that project members for feedback, hopefully next week. And then hopefully we'll send out the actual survey to the masses. So that would include not only our, the PIcc group, but also the TILs working group, as well as other larger entities that have similar interests for this. And so hopefully, we'll have, within 4 to 6 weeks, we'll have, survey data collected, and then 2 to 4 weeks after that, data analysis and interpretation, and then hopefully we can put the manuscript together within 4 weeks or so. And so this is kind of sort of where we are with this, and this is actually all I have to give for this particular entity.
Perfect. Thank you. That's a great overview. Let me just quickly point people. So if you're at the projects team, can you see this browser? Or I hope I'm sharing the right screen.
Yes.
So this is Kim's project, and if you have questions, feel free to reach out, or if you want to contribute, and I hope that many of you will ultimately fill out the survey, so that we have sort of an opinion-based data set to see whether and how this should be regulated, and or how we can provide some input into regulatory decision making. I'll move forward. The next project that I was asked to present, Nisha had to run to one of our classes, is the so-called Seer Cancer Microscope. If you're interested in this project, same route, go to the website, go to projects, find the Seer Microscope. Here are the links. The next meeting is Friday, October 15th. And I wanted to briefly outline what this is, because I think it may be of interest to a broader audience. In the meantime, the group here contains several people. I know Uta and Ahmad may have joined. I saw Uta earlier from Germany in this case. And what this group is working on is using the Seer database, which is a population-based database of the US, to identify over the last couple of decades what the source and modality of imaging is to render cancer diagnoses. And with the help of the entire data analytics team, and in particular led by Ahmed from Ulm, Germany, there is already an implementation here to really screen for individual tumors. Let's pick oral, as an example. This is already a density map of the US and Canadian population, and the percent over the last decades of how frequently was this entity diagnosed with a microscope. Now that may seem obvious to some of the histologists among the group, but as new technologies come up, for example, cell-free testing and others, it is a very important ground truth. And when you look, for example, at tumors of the eye and orbit, that is completely different here. It's not a primary means because there are other optical modalities. In addition, we plot this in terms of a percent microscopically confirmed data across, in this case, 28 very common cancers, just to illustrate. Yes, for example, there are miscellaneous cancers that are not included here, and we are working on a heme versus solid tumor group as well. And here you see, which is a fantastic overview by different respiratory system is portrayed here, but you can basically page through others and compare even across the different versions of Seer and two other publicly available databases. And then finally, sort of to show this trend, basically the preliminary version is that 93% of all cancers are diagnosed using the microscope, which I think is something that sounds simple and intuitive, but demonstrating this formally and into a, I think, publication would be extremely important, especially as some new technologies are coming up, and as I think many on the call here want to change it to electronic and or digital mode. So if you want to join the team, please shoot us a brief email. And again, if you want to join this call, that's October 15th, 9 to 10 a.m. So this is early in the morning due to the time in Europe to make sure that our European colleagues can join as well. So that's a brief shoot out to the Seer Cancer Microscope project. The next project I wanted to briefly outline is the Privacy Pilot Project. And the next meeting happens on Wednesday, October 20th, 3 to 4 p.m. And let me just quickly pull this up here. So the Privacy Pilot Project has also been quite active. You see the current members here. And I think Joe Siren-Trappen is also on the call. I think one of the key elements of this is it's getting into high-level technical details of tokenization. I don't know, Joe, if you want to maybe quickly summarize in 1 or 2 minutes to maybe some of the technical experts who may be helpful.
I wish I had that that PowerPoint that I had. I'm never really prepared to actually show it, but this is in reference to a CAP Today article that talked about the discussion about a national patient identifier, and several folks, in addition to myself, had given an opinion about it. basically we're kind of left to where we started back in the 1990s, where HIPAA started and made a statement that they wanted a national patient identifier for all patients. And it's been kind of a hot topic and a very controversial topic ever since, with basically nothing happening. But it's actually causing a lot of consternation, because in this world where we want data to flow, we want patient records to flow from one hospital to the next, we're being hamstrung because there is a lot of money in the billions of dollars, possibly even higher, about re-identifying patients correctly, and also more so even fraud. You know, there are different ways, because it's so wonky about the way people re-identify patients. Fraud is actually a big issue as well, which I forgot what the numbers are, but they're also very high. So I provided a framework, and I don't have the PowerPoint on here, it might be on the website. Joe, you can tell me if it's on there. But the idea is that instead of doing a national patient identifier, which is for a lot of politicians, in particular, I think, particularly with Ron Paul, he's the one that's actually been pretty much vocally against the national patient identifier for good reason. The national patient identifier, by having one, it's like a social security. Yeah, this is it right here. So this is actually the framework that I had. The problem with the national patient identifier is because it's tied or associated with damaging information, whether you might have a history of cancer or some comorbidity, something that may alter your life because the number is tied to something. Just like the social security number, it's tied to something, financial information, how many credit cards you have. It's the association that's powerful. So, you know, my argument by having the structure here is actually having a centralized server. It's an out-of-the-box solution for healthcare, but not so much an out-of-the-box solution for, you know, technology in general. The idea is that you have a national infrastructure that's able, through tokenization, to identify patients. Its strict purposes alone is just identify. You're not passing an identifier. You're passing tokenization through demographic information that can be tokenized, encrypted, you name it. There are no identifiers being passed through, and sole purpose is to identify whether a transaction is going to take place or not. So if you're a patient going from hospital A to hospital B, this national server, through tokenization, will try to identify through different sets of demographic information. People may not realize that demographic information is not standardized across the nation about how people can identify patients. Well, you don't necessarily need to, you know, standardize everything based on a level of thresholds of different metrics. You're able to actually go to a certain degree of this patient is who they say they are. And through this tokenization mechanism, as well as just centralizing the way people get identified, you're able to identify patients with confidence, and you're not passing any identifiers around, and you're also disincentivizing any hackers to steal anything. If you're stealing tokens, it's meaningless. If you hack the server, there's no demographic information, there's no damaging information like comorbidities located there. It's all within the institutions, you know, it's decentralized among the different institutions. So, you know, it's a way technically to leverage current technology, as well as provide kind of a game theory model to disincentivize hackers to actually be able to do anything with the information that they take. And it really makes it much harder to actually, you know, steal. You have to steal multiple points of entry to be able to do things. So I sort of provided this as kind of an out-of-the-box solution, something that we can look at, and as far as I can tell, it's technically feasible. You know, in terms of it's using current technology. It's just a different way of thinking for a lot of healthcare information security officers. So the resistance might be that this is something that's not seen before used in healthcare, but it's not something totally new. So I don't know if anybody had any questions about that.
So you said something about dynamic IDs. I'm sorry, I couldn't read the.
Yeah, the dynamic ID. So, you know, I said that the national patient identifier is not passed around between institutions, but within the central server, there are probably different ways that instead of having one ID in there, which technically if you hack the central server, you can get one static ID. If you can actually have a dynamic, something that's changing all the time. So if you hack the central server, so what? It's going to change based on some algorithm that you might be totally unintelligible for any hacker to do. You're basically really making it very, very difficult to do anything. And plus the identifier is not tied to anything damaging. That's located decentralized in the institutions themselves. So that's the idea behind the dynamic IDs. You have it so it's switching. I think it's feasible, but like I said, I'm not technical enough to actually design or engineer something like this, but I'm just sort of giving concepts for engineers to design something like this. Didn't know if anybody, I'm looking for people that sort of hack hack my hack, basically, to see, is there anything there? I actually had my chief information security officer take a look at this, and he's a white hat hacker. And he said, well, it's feasible. And I'm really looking to see, is there any argument other than cost and other things, like we've never done this before? That's a stupid argument. You know, there's so many things in life. We've never done this before. Well, tough. Maybe we should do it. If it's feasible, we should do it.
That sounds a lot, Joe, like multi-factor authentication.
I'm not into the weeds about the multi-factor authentication, if they're using tokenization and other things and having a centralized way to do it. But it may be, maybe. But I'm not familiar with how they do that.
Joe, I have a suggestion. So for that discussion, we could use the Wednesday, October 3rd, the 20th date, you know, to continue, because I think there is, of course, a lot of interest in this. But fortunately, we have to move on. But definitely an amazing idea, and I think that requires, you know, continued effort, and we will keep everyone updated. Next topic is the project Test Driving Valid 2021. There is a new meeting coming up October 21st, and I wanted to briefly show what that looks like. So obviously the whole meeting is in here. There was another slide deck here. Let me just pull this up quick. So quickly, for those who are not familiar with the Valid Act, it's like a 245-page document, and it's very, very intricate. And this is the second report on the update of the second session. And we had a first session about a whole slide imaging-based artificial intelligence application to predict combined Gleason grade from H&E stained routine FFPE sections. Basically how we started the meeting and the recent FDA approval. And we went through how that would work under Valid. The second meeting focused on PD-L1 immunohistochemical staining for a solid tumor as a companion diagnostic that is cross-referencing another approved test. So that information is on the website, and how this group thought that may work under Valid. And at that meeting that I just outlined, and you will find the link also on the website, which is October 21st, we're going to talk about how would a next generation sequencing assay that is assessing for multiple established biomarkers work under Valid. So these were the three pre-committed goals. So the next meeting will be basically an NGS assay, but under, you know, from a regulatory perspective. So just in case, you know, this is definitely also up on the website.
So for the third agenda item, are you thinking in terms of like signatures, or?
No, this is more so the basic premise of Valid is that you would have to take LDTs, depending on what risk level they are and what the claim is, through FDA review. And that depends on how the assessment is. So of course, I think a quick summary of that meeting, I don't know if Shannon is on, but was you can talk yourself into high risk with every single assay. And the question is how to make that a little more standardized. And then if so, how would that submission be? Is it a completely de novo, or is it against or cross-referencing another FDA approved test? There are certain, you know, low risk, high risk categorizations. So it's more sort of hypothetically playing what would that look like and how much effort would this be for the individual lab, just to sort of gauge sort of the practical relevance. So this is not about being pro or against the particular legislation. This is more what would this mean from a practical perspective in terms of submitting your validation or performance data to the FDA. Of course. The last item here is something that I think Jitesh has emailed us. This is something from the Medical Device Innovation Consortium. So for those who don't know them, they're a public-private partnership with a lot of industry collaboration in the field of, you know, medical device innovation. And MDIC has launched a digital health initiative with several new work streams. And I think there may be some interest. Let me just pull up the link. So maybe, Ulla, if you can paste this in the chat as well. So this is an initiative to hopefully leverage some of the new regulatory pathways. And I think if you look at these sort of committed goals here, there are several collaborative communities combining their efforts in launching several value streams. The work streams are currently entailing change control, which of course is very key for any AI application, a so-called excellence appraisal pathway, which would be basically a stamp of approval for certain performance and quality metrics. Review determination, which is, you know, sort of like, is there potentially an option to help for digital submissions or submissions tackling digital health problems to have an easier way to identify what kind of review this would be. Streamlining a pre-market review for obvious reasons, you know, to spur it up and facilitate innovation. And then this part here, I think, is very interesting. This is an RWD, RWE, patient-generated data and computer modeling simulation workstream. So these 5 workstreams are currently committed to, and in case someone is interested, please shoot Ulla an email and she can connect you with Jitesh Vitil, who is currently sort of the coordinator on the MDIC side, but we already have committed at least to 2 of these projects, including real-world evidence and real-world data. I think this is the quick summary of what we had on the agenda. So if you have other events, I wanted to quickly point out that if you email that to Ulla, it can be added to the calendar. So we will definitely add, you know, your other meetings and or content to our event calendar. And with that, I open it up for discussion. And I think this time we left some time so we can have an open conversation, ideas, new projects, etc. Silence.
I didn't know if Chris or anybody from Page is on. I just wanted to congratulate about the FDA approval. I sent Joe, I think I might have sent you also the announcement too, but I wanted to know a little bit more about the approval and maybe how does it work? It goes under the de novo pathway. I didn't know, you know, just learning a little bit more about that pathway of how to get FDA approval. And, you know, I was also interested, like, okay, if algorithms get approved that way, as they improve over time, how does versioning happen? All the stuff we've talked about in the past that Brandon has brought up. How do we deal with that? And I'm opening up for everybody.
I mean, we briefly outlined exactly that. I think Carla, he knows their chief business development officer, very, very regulatory science savvy, has just told me that I think the FDA actually posted their press release before Pages, which is, you know, it's like that's fine, you know, because that's the same thing. But I believe some of the documentation is still pending, so you know we're waiting for that. And you know, I've reached out whether they could potentially present to us, you know, what that would entail. But I think it always a bit, you know, after the formal, let's say, authorization notification, until the actual paperwork is released, what that contains, that's also a separate sort of whole process. But once that's out there, I believe many, if not all of the stakeholders will take a look at that. But I think it would be amazing to have Paige present to this group as sort of an expert presentation of what that looked like, and also to draw a line what they can tell us. But just to see sort of how a classifier was assessed by the FDA is highly informative. And, you know, I'm not too familiar with any of the specific sort of domain content, but I agree.
Yeah, I thought it would be interesting to have a blueprint to see how they kind of, yeah, just to see how it works.
Absolutely. Other comments, questions, suggestions? Well, if not, let's end this early, make good use of the time. And thanks a lot for all the updates. And I hope to see you in approximately a month, if not earlier. Thanks a lot.
Take care.
Thank you.